My name is Lenny Gocheva and I'm a postdoc in the lab of Dr. Tyler Jacks here at the KI. I've always been interested in cancer research, and in particular my actual area of focus is the tumor microenvironment. So for a very long time it was thought that a tumor is just a mass of homogeneous tumor cells that proliferate, migrate, invade, and that was the whole picture. However, pathologists for a very long time have known that there are a lot of other normal cell types within the tumor. In recent years, there's been a growing appreciation that these normal cells actually play a very active role in tumor progression. There are a variety of other cell types present, a lot of immune cells and fibroblasts, which are actually the focus of my research right now. So what fibroblasts do, they have a very important role during the process of wound healing. So for example, when you cut yourself, fibroblasts can respond to that and become activated. And what they do is they start secreting factors that they stimulate the surrounding cells to proliferate in order to close that wound. So what actually we're starting to appreciate now is that tumor cells are very smart. They can actually recruit these cells and trick them into believing that they're healing a wound, when actually what they're doing is helping the tumor grow. The image behind me, this was one example of two different cell types that were isolated from a tumor. The red cells, the smaller cells, those are the tumor cells, and you can see there are a lot of them because they're proliferating so rapidly. And then the cells that are in green, those are fibroblasts. And you can see they like growing together. What I'm trying to understand in my research is how these cells are talking to each other. So it appears that it's a dialogue. The tumor cells can talk to the normal supportive cells like fibroblasts, and then fibroblasts can respond in other ways, again by secreting different signaling molecules. If we can understand the interactions between these cells, that can give us a novel therapeutic angle through which we could target not only the tumor cells but also their supportive network. What I've become more and more appreciative of every time I look at images like these and look at slides from our actual tumor is just how complex it is. And I think I'm becoming very respectful of this complexity. You know, a lot of people ask me, why isn't cancer cured? It's just so complex, and every cancer type is so different that really it really takes a long time to understand the role of all the players that are there.